All right, everyone, we're going to get started in a few minutes, just waiting for a few more people to come in.
Okay, I think we can get started now. Good afternoon and welcome to the NINDS Biomarker Q&A webinar. My name is Rakonda Metley and I am the operations coordinator. We will start off with the presentation of slides followed by questions and answers. Please submit your questions in the chat section featured below and they will be answered at the end of the presentation. We ask that you please remain muted at all times. The presentation will be recorded and placed on our YouTube page. I'll be sending that link along with the presentation slides to everyone that has registered. I will now turn it over to Dr. Taylor Birds.
Thank you, Rakonda, and welcome everybody. Thanks for joining today. So I'm Carol Taylor Birds. I'm the program director for the NINDS Translational Biomarker Program. Ram Andrachandran is another program director in our team. He runs the HEAL initiative biomarker portfolio and also holds the pain biomarker grants from the NINDS side. And I'd also like to introduce Dr. Adisa Silu, who's the health program specialist with the group. So I know that we asked a number of questions this time for people who are registering, and I understand that there are a few people who are coming in from a non-research side, and that's great. We love to have you here. I will say this is really geared towards understanding the funding program announcements that we have in this biomarker program. So just be aware that that's sort of the target audience. But happy to talk with anybody if you want to reach out and have additional questions and set up a call at some point. So today we're going to go over just a little bit of background on the program. So the regulatory context for why the program exists, some important terminology and concepts that you need to be aware of when applying, and then we'll talk about the biomarker program funding opportunities more specifically. So each round, we do these webinars twice a year, and each time we try to have one additional topic that we address at the end of the slide set. And this week, or this time, Adisa will be talking about the new NIH data sharing policy because that rolls into effect in January of this year. So everyone submitting to February, this will be applying to you. So it's good to be aware of it if you're not already. So the NINDS Translational Biomarker Program, the purpose of the program is to facilitate the validation of biomarkers and biomarker signatures as fit-for-purpose tools for use in clinical trials to accelerate therapeutic development and aid in decision-making for patient care. So just to put this in context with biomarker research across NINDS, this program is really filling a very specific purpose. NINDS funds a lot of biomarker research across all of our divisions. The majority of the work is done in the Division of Neuroscience, where there is biomarker discovery work, including things like investigating candidate biomarkers and how they relate to mechanisms or pathophysiology of a disease or a condition, phenotyping or characterizing patient populations, and technology development that might be relevant for biomarker detection in the future. The program that I direct is in our translational division. And here we're really trying to focus on biomarkers that can be used in clinical trials and bridges the gap between understanding what the biomarker is in terms of the relationship to the disease or condition or the concept of interest that it measures, and getting them ready to be used in clinical trials. So it's a very focused program. Now, our Division of Clinical Research also has biomarker work in it. They certainly incorporate biomarkers into the clinical trials that are funded, and some of their networks, like the Neuronext network or the stroke networks, also have some ancillary biomarker studies that can come in as part of existing clinical trials. So why do we need biomarkers for therapeutic development. So this is an analysis that comes out every few years by an bioindustry analysis. And it really highlights the fact that having good biomarkers in therapeutic development can really help improve the chance that a trial can move from one phase to another. In particular, in phase 2 clinical trials, you see a marked improvement in the chance to move on to phase 3 when pre-selection biomarkers or prognostic biomarkers or diagnostic biomarkers are being used to help identify the right patient population. Also, overall, you can see that final approval, getting beyond to that final approval significantly is improved with biomarkers as well. So context for the program, so it was developed in 2017, 2018 by Dr. Marianne Pellimonter and a large collaborative group of staff at NINDS. And it was put together in order to help advance translational biomarkers for the neurological conditions. And there was a lot of discussion going on with the FDA and NIH and others in industry. And they put out a paper on, what evidence do we need for biomarker qualification and where are the gaps and challenges. And so this publication highlights what those, what evidence is necessary and sort of the 5 component process for evaluating biomarkers. And so our program and our funding announcements are really built to help develop the evidence in this context. And one of the key points here, and I'm going to just say, in terms of our program, we're saying therapeutic development, not drug development, because really biologics or devices or behavioral interventions are all relevant here. And then the other thing I want to highlight is context of use, because one of the key aspects of demonstrating that biomarkers can be used as fit for purpose tools is being able to define how you anticipate them being used, what the need is, and then demonstrating, developing a study design that can answer the questions that need to be answered for that use case. And this is just to highlight, that again, the reason for the program is that the regulatory process is fairly in-depth, and you can go through a number of stages and may not be qualified as a biomarker or be accepted as a biomarker for a clinical trial by the FDA if there's insufficient evidence. And that was, a key issue that the evidentiary criteria was trying to highlight, and something that our program is here to help with. So context of use is critical to all of the funding opportunities, and I think it's worth pointing out here. When you're thinking about context of use, there are a number of questions to think about. So you really want to be able to articulate how the purpose is going to be used for therapeutic development. When is it going to be used? What stage of development? How is the biomarker applied, right? Is it a standalone decisional? Is it being used with other markers or outcomes? Is it going to be used to increase, the enrichment for certain populations? And what's the intended population? You want to be very specific about that as well. And this comes directly from the FDA's qualification program plan, which you can find online at the link below. So one of the things that we want to make sure everyone's familiar with are the biomarker definitions. And this is the definitions that are in the FDA NIH Biomarker Working Group's best resource. So a biomarker is a defined characteristic that can be measured as an indicator of normal biological process, pathogenic process, or response to an exposure or intervention, including therapeutic interventions. So some other important terminology to be aware of is biomarker signature. This would be a combination of biomarkers or variables or a component or a set of markers used together that would be used rather than a biomarker or a standalone biomarker. So detection method is a catch-all for what the assay or imaging algorithm or analysis pipeline is that's going to actually measure the biomarker. And then concept of interest, I mentioned a couple times already, but this is basically linking what's the biomarker or biomarker signatures relationship to a particular important concept for the biological context. So just as an example, I think most people are familiar at this point with neurofilament light, which is a biomarker being used in a lot of different contexts at this point. And it's one that reflects the concept of axonal injury or neurodegeneration. And then I'll just point out that the terminology for biomarker signature, there's other ones, you could say composite biomarker or multi-component biomarker. And the FDA is also looking at some additional terminology around these. So for the current definitions for biomarker categories, part of highlighting what your context of use is being really clear on what type of biomarker you're proposing to develop and validate. And I know we asked if people were familiar with the FDA's biomarker qualification program and the BEST glossary, and it looked like about 2/3 of people who registered weren't. So I think it's worth going through these. We usually do go through these because this is critical to what you apply when you're proposing your study. So diagnostic biomarker, as you'd expect, used for diagnosis. And I'm just going to run through these, but there's also monitoring biomarkers. So you expect a longitudinal study design for a monitoring biomarker. Pharmacodynamic response biomarkers, these are more often used in earlier stage, phase 1, phase 2 studies. Predictive biomarkers to demonstrate individuals who are more likely or less likely to respond to an intervention. Prognostic biomarker will predict the likelihood of a future clinical event or disease recurrence, often used for patient stratification. And then safety biomarkers and susceptibility risk biomarkers that can also be used for patient enrichment. So the context of use, we went through it a little bit before, but it's a concise description specifying the best biomarker category and how the biomarker will be used in therapeutic development or clinical practice. And examples are to stratify or enrich patient populations, to demonstrate therapeutic target engagement and inform dosing decisions, to indicate risk for developing a disease or condition, to monitor disease condition or changes in response to an intervention, and to inform treatment decisions. And the key point here is that depending on what your context of use is, your study design is going to change. So you need to really think about what you're trying to do and what's the right design to answer those questions. And I covered that one before. So the biomarker validation process and scope is what we parallel in our funding opportunities. And at the discovery and development phase, the first aspect here is to establish that the biomarker reflects the concept of interest, right? Is it neurodegeneration or inflammatory aspect? What part of the disease process or pathophysiology is it reflecting? And then to develop or optimize the detection method to actually measure the biomarker, and then conduct an initial proof of concept that the biomarker can be used for the context of use proposed. With analytical validation, we're then moving on to the next step. You now have developed a detection method, but you want to do really rigorous, robust evaluation of the performance characteristics, preferably across multiple sites and users, and to test if the biomarker or the detection method can accurately measure the biomarker and what the performance characteristics are. So what are the limitations of the detection method? And essentially, can I trust the results of the measurement? And then finally, clinical validation, which is usually large-scale, multi-site studies, where you're going to now expand beyond that initial clinical proof of concept study into a representative large patient cohort and determine, how do you actually statistically address the results of the measurement of the biomarker and turn them into meaningful and interpretable results that can be used for making decisions at the individual participant or patient level. And so these later stage analytical and clinical validation PARs are designed to really help establish the evidence necessary in order to qualify the biomarker with the FDA if that's the intention. And biomarker qualification is a rigorous approach for a conclusion based on a formal regulatory process that within the stated context of use, a medical product development tool can be relied on to have a specific interpretation and application in medical product development and regulatory review. So the advantage here in the pre-competitive space is that having qualified biomarkers can make it much easier to have therapeutic trials go forward with an accepted biomarker rather than having to first provide the evidence that the biomarker is appropriate for how the therapeutic developer is proposing to use it. Now, I will say that for our program, we don't require that investigators go through qualification, but the idea is that the evidence being developed supports the same level of rigor towards showing biomarkers that are ready for use in trials. So these are the funding opportunities that are available, starting with the development. And then we have 2 analytical and 2 clinical validation PARs, one for academics, the UO1s, and then one for small businesses, which is the U44s. So moving now into more specifics about the funding opportunities themselves. So that first discovery and development PAR, 22089, was recently released. So this one is, excuse me. So the preliminary data in our supporting literature should support a strong biological rationale and premise that the candidate biomarker signature or approach for identifying the candidate biomarker may fill an unmet need. So here it's really critical to point out what the gap is in therapeutic development that you need this biomarker for and how it's going to be of added value to existing approaches. And this, again, just to highlight the difference between this and the R01, R21 space, where the division of neuroscience applications would go, is if the interest is identifying biomarkers that are maybe explaining something about the pathophysiology, that's really the R01 space. Whereas if you're identifying a critical therapeutic development tool need, this is where you would be going. So analytical validation then, here the expectation, again, you already have good supporting rationale for it, but you also must have already developed, actually shown the initial proof of concept that you can use the biomarker for the context of use proposed. And you also want to have already developed the detection method, but you can do further optimization in this space. But the method itself, the assay should have some prototype or have some evidence that you've already able to measure what you're proposing to measure. And then finally, clinical validation, adding on to what's already expected in those other 2 PARs. Now you're also asking, should they also have the detection method fully analytically validated? Are you ready to roll it out and know that it's going to be reliable when you're doing your clinical validation study? Then research scope, what can you actually do in the funding opportunities? So for the biomarker development PAR, this is where you can have the preliminary clinical validation. So you want to do that proof of concept study for the concept of use you're proposing. And you might have 1 or 2 contexts of use, and you can propose to test both of them and then move forward with the one that's best performing. You can include the detection method development here as well. You don't have to, but if that's a necessary step, that's within scope here. And then multi-site studies are not expected for this PIR, but they are allowed if that is a need of the program. And then for the analytical validation, Again, this can include optimization of the detection method. Multi-site validation here is expected. We include analytical validation that's appropriate for the context of use. You want to really rigorously evaluate the performance characteristics, so analytical sensitivity, specificity, accuracy, precision, and so on. Evaluate pre-analytical variables, establish the standard operating procedures, right? So basically, what are your sources of variability? How are you going to identify them, quantify them, and address them? And then finally, with clinical validation, here you're establishing the statistical thresholds needed to interpret and make decisions using the biomarker within that specified context of use, so that you have something at the end when you complete your analysis that you can take, you can provide the information how to interpret these biomarkers and the outcomes that you get. So large multi-site studies are expected. Clinical populations should be large and diverse cohorts that represent the context of use that you're proposing. So don't propose something that's very broad and general and then only test it in a small, unrepresentative cohort. And then plans to qualify the biomarkers with the FDA is certainly encouraged, but it's not a requirement. So some basic aspects, just differences between the funding opportunities. So the discovery and development PAR, this is a phased mechanism. This is an R61, R33. Total duration can be up to 5 years. So you split the R61 as either 2 or 3 years, and then the remaining time for the R33 phase. There are milestones that must be included between the R61 and R33 phase, and I'll talk about those a little bit more later. Budget, none of these are limited for the academics, but you need to justify it, and I'll talk about that a little bit more as well. Because you need prior approval if you exceed 500,000 direct costs per year. So intellectual property, that's not a scoreable section, but it should be included, and you should address any constraints, IP landscape, et cetera. So if you're developing an assay that there's already multiple other assays that are commercially available for, that's not as strong of a rationale for why this new one is needed. So you need to consider what you're developing in relation to the outside scope. Resource and data sharing, again, this is not scoreable, but it must be included, and we're going to talk about that more in a bit, because that's now going to be required for all NIH funding opportunities. And then the analytical validation and clinical validation, both of these are cooperative agreements. Again, up to 5 years of funding. The milestones are annual, so you need to include milestones for all 5 years. Again, budget, it's not limited, but you do need to get prior approval if it exceeds 500 direct costs per year. And then the small businesses should rarely exceed 700,000 per year for the phase 1 or 1.5 million for the phase 2. I do want to point out that a lot of the biomarkers that are being developed may not be trying to commercialize their product, their detection method. And if you're not intending to, then you don't need to address the intellectual property aspect, but you should be sure to include how you're going to be sharing your, if it's an analysis pipeline or an imaging protocol, how that's going to be disseminated and shared to the community. So again, this is the phase, I just covered this. So if you are requesting a budget that exceeds 500,000 direct cost, we do need to get prior approval for that. So you need to reach out at least 6 weeks ahead of time, and we'll ask for quite a bit of information that would be also in the application. And that goes through an internal review process at NINDS for permission to come in. Just point out that, some people are confused about the approach for writing the application for the R61, R33. Just to be clear, you do need to write both phases in your application. I covered this already, just additional details here. All right, so finally, we're going to talk about milestones. So what milestones should be included? So you really want to include quantitative performance milestones. So defining the target sensitivity, specificity, accuracy, precision thresholds, and so on, any of those core performance metrics. And that might be the analytical side for the detection method, or it might be the clinical side for how it actually predicts a clinical outcome that the gold standard that you're comparing to. You want to define the acceptable biodynamic range for the context of use proposed. You want to define acceptable variation within and across sites operators if you're doing a multi-site study. Metrics for assessing data quality and assurance might be appropriate. And then, of course, enrollment or sample collection, data collection goals are all appropriate. And then it's also good to include some progress milestones, things like setting up any contracts that might be necessary when you're finalizing your standard operating procedures and how those will be finalized. And then if you're going into FDA regulatory process, meetings with the FDA or submitting a letter of intent to the Biomarker Qualification Program can also be mentioned. So one thing to be aware of, what's not in scope. So I mentioned that the development PAR has been reissued. Previously, it did allow animal work and it does not anymore. So please be aware this is for either retrospective or prospective clinical data samples studies only. And this is in part to help differentiate what's done here versus what's done in the R01 space. Studies that are focused on development of a therapeutic agent or devices as the primary intent are not appropriate. We have other funding opportunities and programs for those. Evaluation of clinical efficacy, effectiveness, or clinical management as the primary intent. Again, we have other programs and funding opportunities for those. Natural history studies that don't specifically work on biomarker identification and validation would be out of scope. And then, like I said, studies using animal models are no longer in scope. And then studies that fail to include milestones, that's grounds for administrative withdrawal. So you do need to make sure you include milestones. Studies outside of the mission of NINDS, of course, are not in scope, although NIA is also signed on for our development PAR 22089, so within NINDS or NIA mission. And just to highlight, these can be withdrawn before review, so please reach out if you have any questions. So finally, just general guidance and things to think about as you're preparing your application. So this is not a payline driven funding program. We take the top scoring applications and go through a due diligence process where we'll ask you to respond to the summary statement. And reviewers are asked, you can read through the review criteria as well as the instructions to applicants and the funding opportunities, the sort of general rules to think about, are you addressing an unmet medical need? Is it supported by a strong biological rationale for why it would be a good, appropriate candidate biomarker, or you'll be able to identify an appropriate biomarker for the context of use? Included carefully designed and needs to be really rigorous plan for performance evaluation of the biomarker and the detection method. Include a plan for standardization of data collection samples for use and validation. Again, thinking about what are your sources of variability and how are you going to deal with them? How are you going to identify them and deal with them? And then provide a strong justification for the utility of the biomarker in the clinical setting or the added value in a clinical research design. So again, how is this going to be, how is it going to impact how studies can be run and how is this filling an unmet need? And then finally, just obviously you need to have basic feasibility. Do you have all of the expertise on your team and all of the resources necessary to actually conduct what you're proposing to do? And just as I mentioned, we do ask investigators to respond to the summary statement if they're in that top scoring range each round. All right, with that, I am going to give my voice a break up here and turn it over to Adisa.
Thank you, Carol. So I think, for these couple of slides, we'll be discussing, in a sense, the data management and sharing policy at the NIH. So obviously, NIH does promote the sharing of scientific data to obviously advance research, discovery, and to make sure that, research results are available to basically the public. And, applicants are and should plan on submitting and sharing any scientific data as they're as they're generating those data. And the scientific data includes any data that is needed to validate and replicate research findings. It obviously doesn't include anything that has to do with laboratory notebooks and preliminary analysis, as well as, any completed, case report forms, but anything in a sense that fits into the validation of the research finding is considered scientific data. When it comes to these plans that are submitted and also the evaluation and the review process, these, I guess the scientific and the data managing sharing plans are not going to be scored as part of the review criteria. So it is merely to have that so that scientific data is going to be made available once the research is completed. So one of the policies that is included, the NIH expects, in a sense, any institutions, PIs that is awarded the grant to not only plan and also budget for the sharing of this data. And as part of that, and as part of the application, you will have to submit a data management sharing plan. That is obviously going to be reviewed by the program director and make sure that it's up to par. And with that, we obviously expect investigators as well as institutions to comply with the approved plans. Next slide, please. So when it comes to foreign collaboration, as we know, a couple of grants could have it. The data management sharing policy is applicable as well, but it's up to basically the investigators, from the foreign institution to make sure that they're kind of familiarized, with the policies of governing the data sharing within those countries and address these in their plan of the application. Obviously, this plan, again, will be reviewed by the program officer to make sure that it is, well thought out. But that is the duty of the investigators to do. When it comes to small business innovation research grant applications, as well as small business technology transfer grants applications, the NIH does accept any withholding of applicable data for about 20 years. But again, you have to state that in the plan as well. Next slide, please. Thank you. Another aspect of the data management sharing policy includes any, model organism or any genomic data as well that is generated. Again, we know that different application requires different, data plans. And with that, when it comes to anything that has to do with organic and genomic data research, we do expect you to include a plan that fits the data that is going to be generated. And again, with that, making sure that you provide a justification for why, some sharing is restricted or even not possible should be part of the plan that is submitted with the application. And when it comes to, in a sense as well, any timelines, we would like the scientific data to be accessible as soon as possible. And that in a sense mean when it comes to the end of the performance period, or that is associated as well with the associated publication of the grants. So it is important that investigators as well kind of keep aware of that and make sure that the plan is followed as well. Thank you, Carol. Some of the repositories as well as the resources that are available, as you can see, we do have the BioSend, which is the NINDS Human Biomarker Biospecimen and Data Repositories. There's the Neurobiobank, as well as the human cell and data repositories that are available to the investigators as well.
All right, thank you, Adisa. And I will say, the last webinar that we did, we also had Dr. Chris Swanson-Fisher on, who runs the BioSend, as the program officer for BioSend, and we will share those slides in this webinars too when we send that out. So that's a resource that you can use either to submit biosamples to as part of your data sharing if you're collecting samples, and it's also a great resource if you're looking to identify samples for a number of conditions that you can use for your discovery or development studies. So before we switch to the Q&A, I just do want to put in a plug too for ROM's program announcement for the HEAL biomarker program. This is a UG3 UH3 mechanism. And he did a webinar recently that is on that YouTube page as well, and you can access if you want more information. And please feel free to reach out to him if this is an area that's relevant for your research. All right, and with that, I'm going to stop sharing and we can switch over to questions. Thanks.
All right, thanks, Carol. And again, those that joined late, please keep yourself muted. If you have questions, please submit them in the chat section below. And this was recorded, and I will be sending the recording out with the presentation sometime next week. Okay, for our first question, for data sharing, is there a specific NINDS repository for various data types not fitting those you mentioned? Is publication of the data enough to meet the data sharing rec?
So there are not specific repositories with a few exceptions. So the traumatic brain injury groups do have a specific repository that investigators are expected to use. Sharing the data, so publication will not be sufficient. The expectation is that you'll be submitting actual the data behind the publications as well. And there's quite a bit of information on this on the website for the data sharing and management plan and policy that Adisa shared. So I'd encourage investigators to look. And you also want to think about the fact of how you're going to budget it, because curated data and sharing data can be costly if you're sharing especially clinical data. So I would encourage you to look through that more.
Okay, next question. Can you please explain more on the natural history studies? What if we are collecting a new type of natural history data with a clinic device specifically to develop new biomarkers for early detection?
Right. So if the purpose of the research, if the research question is trying to validate the biomarker, then that's appropriate and that's within scope. If you're collecting, multimodal data that you're going to do some hypothesis generation on, or you might inform future biomarker identification, but the primary intent isn't to develop the biomarker itself for that specific use case, then it's not in scope. So if it's with a clinical device, if that's a therapeutic device, and you're looking at, a predictive or response biomarker that you're trying to develop with it, again, if the study design is for the demonstrating that the biomarker can be used as proposed, then that would be in scope.
All right, next question. Are you only targeting biomarkers for pharmacological clinical trials, or could a biomarker for an
Suitable.
sorry. Yes, I'm sorry. These are the scientific words that I'm having issues with. Are you able to answer that, Carol?
Yes, I can. Thanks for coming. No, so it can be any intervention or, diagnostic in general. Yeah, no, I mean, it's not, I think as I mentioned before, so we aren't drug specific or biologic specific. It can be devices, it can be behavioral. Any of those are applicable. Absolutely. Other questions? And I may not have said it, but if you do have questions about a specific research project, if you've got a draft aims and want to reach out and set up a call, happy to do that. We do often work with our Division of Translation and Clinical Research colleagues, and we'll have joint calls sometimes if it's not quite sure, what's the appropriate funding opportunity for you, or to get some questions about, get some input on whether it's a good fit for the program. Yes, all of the slides and the recording will be shared. As I mentioned, there's a few extra slides that will be going out to you about BioSend and some links to some of the FDA guidance. And we do these, as I mentioned, twice a year, so there'll be another one late spring.
Okay, we have a new question. Oh, sorry, go ahead.
Go ahead, sorry, Wakanda, go ahead.
Can you please explain in the past what was the pay line, if any?
Yeah, so that's a good question. So for those of you who don't know, NINDS is primarily a pay line driven institution, which means that if you're applying for an R01 or 21, we have a set pay line that's based on the NINDS budget. And so that might be 14th percentile or 15th, 16th percentile. And so applications to regular parent funding opportunities will generally be paid if they're within the top percentile points. With our program, we are not pay line. So this is a special review panels that are held at NINDS. And these are developed as a pay plan. So we take the top scoring applications for each round. We ask investigators to respond with the summary statements. And then we go through a due diligence process with our division team and then our institute directors and colleagues to finalize a pay plan that goes to council for approval. And I will mention, so if you've applied to R01s or R21s in the past, standard CSR reviewers, we generally will have about 3 reviewers. Our program, it's going to be a bit more. You'll have between 4 and 7, because we do have specific expertise that we include to look at particular aspects of the proposals. So just something to be aware of. And this is a panel that covers, well, there's 2 panels, NSDA and NSDB, and these have a broad range of conditions that are represented within these panels. So it's good to think about that because you'll have some disease-specific expertise, but you also have some that might not be as familiar with your condition or have interest. So you can, it's generally a good idea to provide a little bit more context and background than you might in an R01 study section that's, all focused on 1 or 2 conditions. Any other questions? We'll just hold on for a few minutes as people are thinking. And again, do feel free to reach out if you have follow-up questions later. All right, we'll hold on for 2 or 3 minutes just to see if there's anything else coming in. I will put my there are links in the slides that'll be going out as well. You can reach us through the NINDS Biomarker Focus On page. And then our contact information is there as well.
Okay, we have another question. You mentioned there are 3 to 4 other specific area focused reviewers. Can you briefly discuss what those other 3 to 4 areas include?
Yeah, so it depends on the project, but you'll likely have someone who is an expert in modality that you're looking at, right? So if you've got a particular type of imaging or mass spec or whatever your sort of primary detection method approach is, you'll have someone assigned for that. We'll often have a statistician identified, and then just depending on the complexity of the project, we'll fill in additional expertise as needed. And if there are no more questions, people are free to drop off. Again, I'll stay around for a little bit. Thanks everyone for attending. Any follow-up questions? Happy to answer more questions if people have them. All right, if not, I think we can go ahead and end.
All right, sounds good. Thank you.
Thanks, everybody.